So what we presented this year was an interim analysis of an ongoing study we have here at NCI, and its focus is trying to understand which patients with large cell lymphoma benefit from the BTK inhibitor Acalabrutinib. So before our study, it was known that you can safely add BTK inhibitors to chemotherapy up front, and the Phoenix trial, which added ibrutinib to R-CHOP, when that study was published, it showed no benefit across the population, but it did have a subset analysis of patients that were under the age of 60 that seemed like they benefited. And even more recently, our group has published a genetic analysis of these tumors, suggesting that there's specific subtypes of large cell lymphoma that had the benefit from ibrutinib. So it was on the basis of that, we felt like what we needed to know was, the molecular profile of tumors that respond to BTK inhibitors, and perhaps it was broader than we had appreciated. So what we did is we took all molecular subtypes of patients, and we gave all of them acalabrutinib by itself first. So in other words, instead of trying to guess which patients are going to respond, let's actually see in the patients themselves who has tumors that respond, and then we'll end up profiling those tumors later and see if they fit some of our preceding hypotheses. But and then if they respond to the medicine, then they continue with that medicine with chemotherapy. But if their tumors don't respond, then they just get the standard chemotherapy alone. So this window design answers a lot of questions. And the reason we reported it early before the results are done is because a number of our patients that have tumors that respond actually have subtypes of large cell lymphoma we would have predicted probably don't respond to acalabrutinib. So that was one of the major findings. And then the other major finding was that in looking at the ability to predict response, what we saw was that really early changes in circulating tumor DNA as early as a week actually were pretty tightly correlated with changes on CT scan. And so that's consistent with what we've seen across other studies too, that the changes in ctDNA are very early and they're rapid, and they seem to correlate pretty tightly with clinical response. So those were kind of the main important take homes from that.